overview. So it's going to be a fairly broad and simple lecture to just give you an introduction to the immune system. And this is what we're going to discuss. So first we're going to look at the different components of the immune system, what makes up the immune system. Then how does the immune system recognize the enemy? Right, and that's a really important question. It's not so straightforward actually. How do the cells of the immune system and the mediators of the immune system get to the site of the infection? And then we're going to look at the activation of the immune system. And your immune system is obviously really crucial because us all as multicellular organisms that have a nicely controlled metabolism, nicely controlled heating, there's loads of nutrients around, right? We're just a perfect place for opportunistic pathogens to live. And so in order to survive, and for all of us to sit here this morning and get here despite land transport, it's really important that we can keep invaders at bay. Also that our immune system, because it's a very, very powerful protection system, but also we have to make sure that it doesn't turn itself on us, that really we know what is self opposed to what is foreign. And that for a long time sort of was a bit of a conundrum how this would happen. How does the immune system know? Because as you probably are all aware, if your immune system doesn't make the distinction correctly, then you'll end up with disease as well, right? So then you'd have autoimmune disease, which can be really devastating, or you develop allergies. That's this is a reaction to foreign, because the allergen isn't part of yourself, but it's the reaction to something that is completely harmless. And so really, how do you make these decisions? That's a really important question to answer. So first, we're looking at the components of the immune system. So in technical terms, what is the immune system? It's a system of cells and molecules that work together to fight invading pathogens. But really, maybe this should also be extended slightly now that I'm looking at it. It should also say it also fights to protect us from cells of ourselves that have grown to be something else than they're meant to be, like cancer cells. Because the reason that we don't all develop cancer all the time is that the immune system also keeps that in check. So if one of our cells starts doing something it's not meant to do, starts proliferating massively, uncontrolled, or changes its phenotype to a completely different cell type, it would be recognized and then killed. And as with a lot of things in biology, you'll find there's always some sort of, we like to put things into little boxes and call them, this is the box innate immune system, this is the box adaptive immune system. And the more you start to study all these subjects, cell biology, molecular biology, medicine, you'll find really these boxes are quite artificial, and really all these systems really work together. But to get a bit of an overview is quite helpful. So the innate immune system, the more simple and the older immune system we have, and you can also find it in worms, in vertebrates, and it's very broad. It always stays the same over the lifetime of the organism, so you're born with it, and when you die, you still have the same system. It's germline encoded, so it's mainly the same for all of us. And yeah, as I said, it already can be found in simpler organisms like plants. And then we have the adaptive immune system. That's quite a different matter because that is highly, highly specific. It can adapt to your environment. It's produced by gene rearrangement, and it's only found in higher vertebrates. Now, however, these two systems, they're closely interlinked and they're closely linked to work together, and the one does not work without the other, especially the adaptive really needs the instruction by the innate immune system. And although there are cases known where people have defects in the adaptive immune system, if you were born, for example, without a phagocyte, which is a macrophage, basically one of these huge cells that can ingest macrophages can interact with cells from the adaptive immune system, the lymphocyte, the T cells, the B cells, and there are people with T or B cell deficiencies, and some autoimmune disease, for example, rheumatoid, can be treated by artificially depleting all the B cells, because this is what's causing the disease, and you can then still survive and still get antibody replacement therapy and still be healthy. And these are a few of the cells that make up these two different arms of the immune system. And really important, I think, I wanted to put here in little brackets. So these are the group of the phagocytic cells, and one of the most important ones is the dendritic cell, which directly instructs the adaptive immune system. Then we have the phagocyte, the macrophage, the monocyte, neutrophil, eosinophils, and can fight infection. Then you have basophils, and platelets, and erythrocytes, which also have important functions, not as often discussed in immunology, unless you're talking about disease such as allergic reactions, where basophils is important. And then we have the adaptive arm. And then we have basically the T cells, and we'll talk about them a little bit today, the B cells, so these are your two very, basically the very famous arms of the immune system, B and the T cell. And then we have the natural killer cells, and with these cells, it's a little bit of a question, sort of, they sort of sit somewhere slightly between these two groups, because they've got a few traits that make them very much like adaptive cells, and they can kill infected cells, and they can also, they're very important in tumor immune surveillance, while also they have a lot of innate features, so they're a little bit of a in-between cell, and probably they are a little bit older in evolutionary terms than these cells of the more highly developed adaptive cell. Now, if we had an infected cell, right, so this is this long epithelial cell down here, and produces a lot of virus, and they get some virus inside this cell, but also some virus will be released. And so for this, we have two different cell types that can deal with it. So T cells, they can recognize things that are inside the cell, but they cannot recognize natural protein. Everything that they can recognize needs to be presented to them. And then we have the B cell up here, which can produce antibody, and the antibody can bind to, for example, virus or bacteria, other things that are not intracellular pathogens, but that are outside. So once the virus is released from the cell, it becomes vulnerable to being bound by antibody. However, these two arms of the immune system, they cannot act by themselves. They don't get just activated by seeing things. They really need to be instructed. And this is what's the work of the antigen-presenting cells. These can be dendritic cells or macrophages, which belong to the innate arm. And they really need to activate the T cells and the B cells, and there has to be an interaction between T cell and B cell in the activation in order to allow these arms to work. So you can see that there really is a very intimate contact between the two arms, and we need both sides to work efficiently. Now, antibodies we've already talked about, and antibodies are something that obviously are super important because they're very flexible. They can bind a billion different molecules. You can engineer them very nicely. Obviously, antibodies is what you produce when you get vaccinated, for example, and all our vaccination strategies, they all depend on antibody production. And if you're not producing antibody very well, you have recurrent infections. And also, they're one of the pet molecules of basically every life scientist. Because we can use antibodies to to find specific targets. So for example, we can raise an antibody to a T cell, and this is how we know if we have some blood, whether there are T cells in there or not. Or we can raise antibodies against HIV, and this is how we can test whether a person's infected. So antibodies, we can bind to the antibody fluorochromes, so then we can make things glow. We can bind toxic compounds to antibodies, that's being done now, and then antibodies are being raised to, for example, tumor cells, and the antibody specifically then delivers a toxic compound to the tumor cell, killing the tumor cell, not killing other cells. So they are also really, really important tools for therapies, and they're being more and more used. And we've got a lot of different antibodies in our labs that we use on a daily basis. So these are obviously not only naturally important molecules, but also as tools for our work as scientists. These are really invaluable. Then we've got the complement system, quite an ancient system, and we sort of, I think for a really long time, at least when I was sitting over there, I always thought, oh my God, complement is like the most boring thing in the world. So the minute I'd written the exam, I completely forgot about it again. But actually, complement now, we start to understand that the complement system not only complements the immune system and stimulates inflammation, but also does a lot to modulate the immune cells themselves, to modulate the cellular metabolism, to make immune cells more active against pathogens, to really make them cope in special environments, because you also have to think that a cell that maybe runs through your bloodstream is in contact with a lot of oxygen, so it's very easy for it to take up oxygen to fuel its energy, but when it goes into an organ, especially something like a tumor, the oxygen tension there can be extremely, extremely low. And so our cells, or the amount of glucose can be extremely low. The amount of amino acids can be low. So it's really important that the cellular metabolism adapts very quickly to different environments to allow the cell to survive in a place and function in a place that is probably sometimes quite hostile to it. And actually, you'll probably hear about this a little bit further on in your course, how tumors, for example, use that and deplete the environment they grow in, basically their own environment, from things like glucose. And this really suppresses the immune system. So metabolism is now one of the very big growing fields in immunology that we're all looking at. And then we've got cytokines. And cytokines are little molecules, and they allow one cell to communicate with another cell. So they can use cytokines, and let's tell one cell, oh, you have to switch on an antiviral program, or come here and help me, I think something is wrong, or it could say something like, something with you is wrong, mate, you really need to now switch off and go into apoptosis, which we call the programmed cell death. So the cell with an infected cell could, for example, be induced to kill itself, and therefore it will contain and stop the infection. So cytokines are really vital in immune cells to communicate with each other. And the body cells, not only the immune cells, but all the body cells, because conceivably it's not normally your immune cells that get infected by viruses or by other pathogens, but it's often all your other cells that get infected. And these cells can then signal. And then we've got adhesion molecules. And that's really important for cellular organization, because your cells move through your body now. If they want to stop in a place where there's, for example, inflammation, then somehow they have to hold on to the endothelium where they are to stop moving and to then remain in that environment in order to have some effective function. And then we can, of course, produce microbicidal compounds that can kill microorganisms directly. And they're right component. And as I just mentioned, apoptosis will kill not necessarily the pathogen, but they will kill the infected cell. And at least for things like viruses, obviously if their host cell dies, the virus can no longer replicate, because the virus itself is not in that sense alive that can replicate itself. It needs a host always. Now, we come to the question. We've got all these nice components, and some of them are clearly quite dangerous, because if you think about it, if you've got a cell, and this cell produces a molecule that can tell other cells to kill themselves, then it would be really dangerous if that molecule was randomly released, because what you get is just some massive inflammation. And actually, cells that can change the vascularity, so it can make your vasculature more leaky in order to move from one part to the next. Because if they want to move out of your vasculature into inflamed tissue, this place becomes slightly more leaky, and then immune cells can move in. Now again, that on a small scale is a very important mechanism, but on a large scale, that is hugely, hugely dangerous. And if you think about it, patients where this can be seen as an acute sepsis, where people have a massive uncontrolled immune regulation, and suddenly their blood pressure drops, they have this huge massive inflammation in all the organs, all the vasculature becomes completely leaky, cells start dying, and that can be a lethal condition. So really your immune system has to be super, super controlled. And therefore it's really important that we don't just activate the immune system randomly, because it's your friend, but it's also your foe. So, but it needs to be only then activated when we see a pathogen. And in immunology we call pathogens also antigen, so the targeting. And in order to recognize your pathogen, the immune cell needs to have a receptor that is specific for things that are only found on pathogens, not on your cell. And this is one of the ways it can act. Or there can be immune system molecules that are soluble and can therefore move a bit further, and they can bind to the pathogen, and then the immune system cell can bind this molecule and therefore recognize the pathogen. So that's a good way if this immune system cell does not have a specific receptor for that pathogen. Because you can think that the binding site here could be completely variable. As long as the site that binds to the immune cell is always conserved, the immune cell can basically recognize an infinite number of pathogens by this pathway. And it doesn't need to have the correct receptor to see the pathogen directly itself. And that's basically how an antibody works. And so if we look at phagocytes, so the innate arm, which have receptors on these cells that are specific for a molecule which is associated with several different pathogens. So a lot of pathogens, like bacteria and viruses, they use structures that we don't usually have in our own cells. That's usually more true for fungi and bacteria. While viruses, because they use our cells to replicate, obviously they cannot make any molecule that's completely foreign to us that we don't make. They can have structures that are not the way we would have a structure in our body usually, but they need to use the material that we basically provide, because they can only use whatever the host provides for them. But of course, if you look at virus under scanning electron microscopy, it's got usually lovely, very neatly built structures, and this isn't something you find in a mammalian cell. So examples would be, for example, a receptor that can recognize bacterial flagella, so a component of the flagella, and no human has got flagella anywhere. So that's quite a safe thing to recognize. You know, this is something we don't have. Now for lymphocytes it's a little bit different, because the receptors are very specific to a unique given pathogen. So a T cell or a specific T cell or specific B cell will always only recognize one target. So maybe you've got a T cell that recognizes a little part of the core protein of HBV, of hepatitis B. That's it. It will not recognize anything else. This specific T cell can only recognize this one target, and all its daughter cells will also only recognize one target. However, you've got a lot of T cells and a lot of B cells, so in the entirety, they can recognize a lot of different things, but each single cell only recognizes one target. And for this, the T cell has got the aptly named receptor, T cell receptor, or TCR, while on the B cell, it's called the B cell receptor, and basically that just means it's antibody that is bound to the B cell surface and being released. And here we've got this, so here we've got antibody on the B cell, which is stuck to the surface of the B cell, it can signal to the cell. And in the case of the T cell, we have the T cell receptor, and we've got two different types of T cell in the body. So one that uses an adapter protein, which is called phol, and therefore we call this T cell the CD4 T cell, and this T cell here is the CD8 T cell because it uses an adapter protein that's called CD8. And these adapter proteins, they aid the binding of the T cell receptor. Now, so we can recognize the enemy because we've got specific receptors that are just against the pathogen. Now, once we've done this, of course, the cells need to get to the site of infection. And this being done, cells are being recruited by cytokines, so they get signals, and because there's a gradient of cytokine, they are moved towards the gradient, towards where the cytokine gets stronger. And then the cell needs to start adhering to the endothelial cell when it's in the vasculature in order to move into the infected tissue. Now, you'll probably come across these if you look at an intravital microscopy, so where you film cells moving in the vasculature, for example, like mice. You can see these cells, they really speed through the vasculature quite fast. So if you think there was like a little infection here, and the cell comes along, it will just zoom past, basically. So really, it now needs to be slowed down, and this is being done by adhesion molecules that the cell can bind to. And the cytokine the cells send will induce the cell to express these adhesion molecules. So it will start making loads and loads of adhesion molecules. The endothelial cells here, they can sense, because of the cytokine, they also sense that there's infection going on. They produce the corresponding receptor, and then the cell can start adhering. They slow down, eventually they stop, and then they can move into the tissue, and they can remove whatever invading pathogens there. And then, finally, we want to, of course, know how the immune system is then activated. So it moves into the system, into the vasculature, then it moves out into the underlying tissue that's inflamed, and there's pathogens there, and now we need to activate these cells properly in order to fight the pathogen. And as I've told you, it's really important to recognize what is self and what is foreign. And the two people who really pioneered this work are these two shown here. This is Charlie Janeway and Polly Mattinger. And the two of them, Charlie, unfortunately, already passed away, but he's left a really, really brilliant immunology textbook, Janeway's Immunobiology, that I'd highly recommend to anybody who's interested in immunology. It's really good. And it's still being updated in his name, but obviously no longer by him. And he's proposed that actually what what the immune system sees is the immune system gets educated what is self. And this is especially true for the adaptive immune system. So our adaptive immune system gets educated what is self, and if something isn't self, then it's what you call non-self, and that is potentially dangerous. And that was a nice sort of system in the beginning, and people thought, oh yeah, this is brilliant. So you either have self, and then the immune system doesn't do anything, or there's something that looks foreign, and now the immune system can respond. However, that of course is exactly the case if you think about it in allergic reactions, right? So a pollen clearly is non-self, but should we respond to that? I mean, it's not dangerous to anyone, right? It's completely harmless. And those of you lucky enough to not have any allergic reactions, I mean, you're all really fine. And actually, this completely aberrant response of the immune system makes us more sick than it helps us. So really just saying the immune system needs to recognize non-self is really not good enough. And this is, I think, what Polly then really recognized. And she then went at a conference and spoke and said, well, really, what the immune system also needs to recognize is not only non-self or foreign, but it also needs to see danger. And this is how the danger hypothesis came along and was coined. And so there has to be changes that the immune system sees that says, okay, there's something foreign and this is dangerous. So there's either some damage to our own cells, and they then respond to a stress signal, or there's some pattern recognition receptors that are never good because they are specific to bacteria. Maybe you find something in a location where it's not meant to be, because bacteria by itself also, that is not necessarily a danger. Our intestine is completely full of bacteria. Our skin is full of bacteria. Actually, standing here, I've got more bacteria cells than I've got own cells, right? So I'm more bacteria than myself. Because bacteria is so teeny tiny that you're completely covered in that. Your intestine is completely covered. Everything is full of bacteria, so basically you're carrying around more little hosts on yourself than you're yourself there. So, and that's not dangerous to you. And it's also really important because germ-free mice, where you take away all the bacteria and everything, when they grow up, they start developing disease. They develop all kinds of autoimmune disease, and they're really not very healthy. So clearly, the biomass we're carrying with us has also some really important functions for us. But of course, bacteria in your intestine is the one thing. Bacteria, for example, in your bloodstream is really not very good. So it's also often about the location where you need something. And so this danger hypothesis basically said it has to be some way that the immune system recognizes this thing isn't meant to be where it is, and therefore it needs to be tackled. And this is exactly what's true. And this was a big publication for her as well. And famously, I think this is one of his pictures here also with a dog. She had developed all this idea, everything, and had this list of herself and I think one or two other authors. And then she thought, well, somebody who's really supported me basically, or helped her develop the idea, I have no idea, is her little dog, so she put the dog on the paper as well as a co-author. And the journal wasn't happening. When they found out that one of the co-authors actually is a dog, they were really, really unhappy. And I think since then, many journals will always ask you what the contribution is. And I think sort of, well, taking me for a walk or something doesn't really count. So yeah, so there was a huge outrow as one of these. So she's quite a character, actually. engulf. Here we have like bacteria, the phagocytic cells can engulf this, take it up, and then once the bacteria is inside the cell, which could be a virus or any kind of foreign antigen, it can be destroyed within this cell. And for this, it can recruit different molecules and then destroy cargo. And this is why it's really important if you've got lots and lots and lots of phagocytes in your lungs, for example, and they constantly move around your lung and clear your lung from everything that we inhale the whole day long. Because there's, of course, a lot of spores and fungi and bacteria, viruses, all of that. Another place that actually has the largest population of phagocytes in your body, what do you think is that? Any guesses? Come on, all of those of you who like to go to the pub, what could it be? Yes, the liver. Yeah. The liver has got a huge population of macrophages, and it's really quite important because a lot of the blood of the gut drains into the liver, and there we take up a lot of our nutrients, but of course there can be a lot of byproducts from the gut as well that needs to be broken down and dealt with, and this is why we've got a lot of macrophages there, and they're called Kupffer cells. Now, as we've already said, the adaptive and innate immune system work together. And one thing that happens, so the B cell can actually also take up some bacteria or other products, pathogenic products, but it can also produce antibody. The antibody can then coat a pathogen, and macrophages can bind to antibody and then take up whatever the antibody has bound. And this would look like here. So the antibody has been produced by the B cell, can bind to a microbe, and all the macrophage needs to have is a receptor for this antibody, then it binds antibody. And many of the receptors of the phagocyte on the same side of the cell bind to something, then this automatically triggers engulfing of whatever it is that's done. And then, so this is how the innate immune system afterwards, the macrophage can then move on and then find the next bacteria and use that one. Actually, macrophage just means big eater. So macro is big and phage engulfing. Now, for B cells and T cells, the thing is you have, as we said, you have not a huge population of B or T cells that recognize the same pathogen. So when you get infected by something like flu, then you might only have very few B cells that can produce the right antibody that recognizes the flu, and you might only have very few T cells that can recognize a specific virus. And that means that upon activation, what these cells will do specifically, because they've been activated, because they recognize something, they start to proliferate. And we call this clonal proliferation because they can only give rise to daughter cells that are exactly as they are themselves. But it means that you, at least for a little while, increase the population of those cells that are specific to flu to a very, very large extent. These cells can replicate very fast. And then you'll have a lot of cells that can produce flu-specific antibodies because they've now all come from this one cell that has been activated there and starts to proliferate. You've got more and more cells, they all produce the same antibody, and you can combat the flu. And the same is true for the T cell. This T cell has been activated, starts to proliferate, and now we've got a huge amount of T cells that can all recognize flu and all combat it. And because this is one of the major functions of the immune system, of the adaptive immune system, that when it gets activated, time you only have a few of these cells, and when they get activated, they start to proliferate really, really fast. This is why chemotherapy, for example, is so devastating for people, because these cells proliferate so fast, so they get killed along with tumor cells, because the target cells that are fast proliferating. Now, once you've got all these cells and your pool is combated, then most of them will die, but a few of them will remain, and this is what gives you long-term immunity. And that's why if you've had whatever measles as a child, you're not getting infected again, because these cells will stay around for a very long time. The next time the same pathogen comes around again, it will be recognized much faster, cleared basically immediately, and usually therefore you're not even developing any symptoms. And this is what's giving us the long-term immunity. Now, today we've basically looked into cells of the immune system. We've learned that they have receptors for pathogens. They need to get them to the site of infection, because not necessarily they will always recognize the pathogen at the site of infection. But this can disseminate, like in the case of viruses, for example, hepatitis B virus or C virus is produced only in the liver, but the whole body is full of the virus. So you can find viral compounds everywhere, but the actual infection only happens in one single cell type in the liver. So even though your T or B cell might recognize the pathogen as something completely different, still needs to get to the site where the pathogen is probably being produced. Then they need to be activated to carry out their function, and the broader the range of functions is, the better it is for the destruction of pathogens. So we know that if we have something with a very narrow focus of the immune system, then the clearance isn't very good. And this is, for example, the case in people that naturally resolve HCV, so hepatitis C. We know that in them they have a lot of T cells that have recognized the virus, a lot of B cells that have recognized the virus. We have a lot of different parts of the virus that can be recognized, and they naturally clear. If, on the other hand, you have quite a narrow response, where you have only a few clones that have recognized the virus, and they're only directed against one or two epitopes on the virus, so a little bit of the antigen, then it's also much easier for the virus to mutate that little part and then still hang around. So then the clearance isn't very good. So a very broad response where lots of different cells from the immune system, a lot of different molecules are involved, is always superior. And finally, this is a little list of recommended treatments. So if you're interested in immunology at this stage, I would really recommend to you to have a look at this, how the immune system works. It's a small book, and it's a really good, simple introduction, brilliant overview, it gives you a nice foundation, and I think it probably covers everything that at this stage you would need to know. And it's easy and nice. Then Janeway's Immune Biology is a classic. I've already learned with it, and it's always been basically a friend, brilliant book. And then I think Pete Bells is going to give the lecture tomorrow on Wednesday. And he's one of the co-authors of the Roy's Essential Immunology. It's a lovely textbook, but it's a bit more scientific. It explains experimental concepts very well, but it might be that at the moment you might feel it's a little bit advanced. So this might be more something for the years to come, while I think the first two are probably slightly more easy.